Okay. Not because I know anything about biophysics, but because Marla was my favorite former student. Not because she is a great scientist, but because she is a wonderful, nice person. Marla was from our own department. She graduated from the top of her class and received the department's citation. She continued to do graduate studies here, and I was fortunate to have her join our group. She did great work in laser spectroscopy and liquid crystals. After PhD, she went to Bell Labs, and she had the courage and vision to jump into biophysics, where she didn't even know what a cell was. She came back to Berkeley as a Miller Fellow to learn real neurobiology from Professor Carla Schatz. Afterwards, she went to NIH as a research staff member and to UC San Diego as a faculty member. She was recruited back to the Department of Molecular and Cell Biology at Berkeley in 2005. She served as the chair of the Division of Neurobiology for a few years and is currently the Paul Licht Chair Professor, Distinguished Professor in Bioscience, and the director of the Miller Institute on campus. She has received numerous awards, such as Brian Boycott Prize for Career Accomplishment and Pioneers in Neuroscience Award. I want to specifically mention that she is a recent winner of the Faculty Mentor Award and the Distinguished Teaching Award on campus. She is obviously a great speaker and lecturer. Marla's main research interest is in neuroscience on how neural circuits are wired up during development, which is the topic of his colloquium today. Marla, please. Marla?
Yes, yes.
Okay.
I am here. All right, well, thank you very much for that very generous and accurate introduction. That was nice. It is true, I did get my undergraduate degree and my PhD from the physics department at Berkeley. And it is, of course, a great honor and a little bit terrifying to give a seminar in front of one's home institution, particularly when you're a biologist giving a talk to a physics department. But the landscape has changed in the physics department since I was a graduate student, and it's very exciting to see the kind of diversity of research that's going on now in the department. And I'm looking forward to talking to you about the project that one of the projects we're working on in the lab now. But I'm actually going to give a fairly extensive introduction because I know that there's not a lot of expertise in neurobiology in the department. I should say that I am not a biophysicist, as Ron did say. That was I actually am a neurobiologist, and because I know there are many biophysicists out there, and I would not want to step on their toes. But also to sort of make clear sort of what the motivation is for the experiments that I'm doing. So I'm interested in studying vision, and I want to understand when one says they're studying vision, it means that they want to understand how when a neurobiologist says it, how the brain represents sort of all those photons that come into your eyes that are scattered off to objects in the world. Now, the animal that I choose to do this research in is a mouse, and I want to start by removing the thoughts that you might have right now that say that mice can't see. So what you're looking at here is a mouse that is tracking a cricket. The cricket is here, it likes to eat crickets. It is doing this completely with its visual system. You can plug its ears. You can put the mouse, or you can put the cricket behind an acrylic shield so that the mouse can't smell it, it can't touch it, and it's able to use its visual system in order to track its prey. And so mice are not blind, and they are the animal that is the that I will be talking about today when I talk about vision. So a goal of people who do sensory neuroscience, so in my case, I want to understand the sense, the visual system, the sense of sight, is to understand a neural representation of the external world. And to give you a sense of how complicated that is, what you're looking at here is a camera that has been put on the top of the head of a mouse when it's in a cage. And these are all the visual signals that are impinging on the visual system of this mouse. And somehow this information has to be encoded. And what you're looking at here is an array of neurons. And when they flash, they're responding to visual stimuli. So the goal of sort of my field of research is to try to understand how the brain represents this complex visual scene in this pattern of activity in these neurons. So before I get, and I should say that my lab, I meant to say, studies this in the first part of the visual system, which is the retina. And actually this movie you were looking at was that of retinal neurons. But before I get to talking about the research in my lab, I just want to give a little primer on how signaling works in the nervous system. So the way that I, sort of the scale of which I like to think about problems in neurobiology have to do with what we refer to as neural circuits. And neural circuits are comprised of individual cells that make up the brain, which are referred to as neurons, and the connections that they make. So let me just introduce these concepts. So a neuron is a single or individual cells, and these are the cells that make up the brain. And neurons are polarized in that they have an input and they have an output. And that polarization you can see not only from their shape, but also by their function. So the shapes of the inputs to the neurons are these little tendrils that are coming out. So this represents the cell body. And here are these tendrils that are coming out, and these are referred to as dendrites. And that's where the inputs come into the neurons. So those dendrites will either be touching somehow the external world, or they will be getting inputs from other neurons. That information, so that's the input side. The output side is this incredibly long extrusion of membrane that comes out of the cell body, which is referred to as an axon. Axons can be very long. They can be up to meters long and still comprised of the same cell. And all of the information that comes out of a neuron is carried down this action potential. It's carried down this axon, and there it makes a connection with another neuron, which is the receiving neuron, and it will make that connection with the dendrites of that neuron. So the output of this neuron, the axon, the terminal of that axon, will form a connection with the receiving dendrite of another cell, and that structure where that connection is made is referred to as a synapse. So the synapses are the connections between the neurons. Okay, so those are the components of the circuit. Now, how do they signal? So cells are amazing. As Ron said, I didn't really understand what a cell was when I got my PhD, but now I understand very well. And one of the things that's interesting about cells and neurons in particular, so cells are, this is all one single membrane. They're surrounded by a lipid bilayer. And that lipid bilayer serves as a capacitor so that there's charge, that the membrane is always charged. So there's always a potential across these neurons. It's a battery, they function as a battery, and the potential across that of the membrane of a neuron is around 70 millivolts. So it's that takes energy to maintain that potential, and that's sort of why the brain uses so much energy, because you have to keep these batteries charged on all of these neurons. So that's going to be our source of energy that we need for the electrical signaling that the neurons do. So as shown here, so now we want to talk about what those signals are. And one of the signals that travel down the axon, the output of the neuron, is called an action potential. And in this slide here, I explain what an action potential is. So again, we're focusing on the output of a neuron. So an action potential, the output of a neuron, is an electrical spike that is actually kind of a phenomenal thing. So an action potential refers to a change in the membrane potential of around 100 millivolts, and it lasts for about half a millisecond. So it's a very rapid and large change in membrane potential. And interestingly, it's an all or none event, which means when that membrane potential gets excited to say around minus 50 to around minus 55 millivolts, right? A threshold is reached, an all or none event happens, and an action potential is generated. In a given neuron, that action potential is the same size and the same duration, no matter what the circumstances. So it's kind of a binary code that's generated by this neuron. So if that neuron has a stronger stimulation, it doesn't change the shape of the action potential, but what it changes is the frequency of those action potentials. So a strong stimulus will give you higher frequency of these electrical spikes, and a weaker stimulus will give you a lower frequency. So intensity of stimulation is encoded by the frequency of action potentials of these all or none events. Okay, so now where does this excitation come from? Well, the thing that excites a neuron, as I just showed in the last slide, are inputs via synapses from other neurons. So those synapses come in two categories, two flavors. They either excite the cell or they inhibit the cell. So if the synapse that comes in excites the cell, if it's excitatory, then it brings that membrane potential closer to threshold. And if it's an inhibitory synapse, it brings that membrane potential further away from threshold. And so the job of this neuron is to integrate all of its synaptic inputs, done mostly in a linear fashion, sums them all up. And if there's enough excitation, it'll reach threshold and an action potential will be fired. Okay, so neurons generate electrical events called these action potentials, and the synapses provide this excitatory and inhibitory input to neurons. So it seems like a very simple scheme. However, it ends up being very complicated when you look at the number of synapses that are in a given neuron. So what you're looking at here is one of the most common cells in the mammalian brain, which is called a pyramidal cell. It's found all throughout your brain. And what's labeled on this pyramidal cell are all of the excitatory synapses and all of the inhibitory synapses. So this neuron, when you're looking at the cell bodies there, you're looking at all of its dendrites. Its axon is coming out at the bottom here. So these are all of its dendrites. It receives 10,000 excitatory synapses and 1,000 inhibitory synapses. And all of that information gets integrated by the neuron, and it's converged, and its output is in the number of action potentials that are fired by that neuron in the end. So the output of a neuron depends on the integration of the synaptic inputs, and there's also a role that the electrical properties of the neuron itself plays. Okay, so now that you have all the tools you need to understand a neural circuit, let's get to the circuits that I care about. Okay, so I like to study this question about how neural circuits perform computations in the retina. And I think of the retina as a really tractable place to address this fairly complicated question. So let me introduce you to the retina. So light scatters from objects out in the visual world. You have a lens in your eye that focuses that light onto this sheet of cells in the back of your eye. That's referred to as a retina. Now, the retina is made up of multiple cell layers, and here we're going to look at it in cross-section. So light actually passes through the tissue of the retina. So the retina has some thickness to it. Light passes through that tissue, and it activates the cells at the back of the retina, which are referred to as photoreceptors. So these are the cells that have specializations that absorb photons, and it converts that signal of absorbed photons into these changes in membrane potential that the neurons use to communicate. So they're at the back of the eye, okay? So that's where vision starts. There's then processing of that information by a series of neurons in the middle of the retina, and it's these series of neurons that are somehow extracting the critical bits of information from the visual scene. And that extraction of those features that are important in the visual scene get encoded in the output cells of the retina, which are referred to as ganglion cells. I apologize, here are what they're called. So these are called ganglion cells. The ganglion cells are the output cells of the retina. They fire action potentials. Their axons make up the optic nerve, and they carry all the information about the visual scene up into your brain. Right, so it's amazing. You start with these photoreceptors that are probably linear reporters of luminance in all parts of the visual scene, And then a couple synapses later, you have cells that are firing this binary code that is all the information that your brain is going to get about the visual scene. Okay, so there's definitely a lot of extraction of information that's happening in this process. And that extraction is happening through these neurons that are in the middle. Some are excitatory and some are inhibitory. So the diversity of computations that are carried out by the retina, and by that I mean the sort of different features that are extracted, can be represented essentially by the numbers of cells. So let's say if we start at the photoreceptors, there's actually only a couple different kinds of photoreceptors. There are rods and there are cones, and you may have heard of those. They're the ones that the rods are sensitive to very low photon flux, and the cones are better for daytime vision. These are these neurons, excitatory and inhibitory neurons in the middle, and the ones I want to focus on are these output cells or the ganglion cells. So there's estimated to be essentially 60 different types of ganglion cells. And from that, we would say that they encode 60 different features of the visual scene. So the feature that we're most interested in the lab is that of motion, of things moving in the visual scene. And that's something that our visual systems care about very much. That mouse could not catch that cricket if it could not sense objects moving. And there are many of these ganglion cell types that are sensitive to motion. So I'm going to flip the orientation. We're looking at things in the cross section of the retina, but we're going to flatten the retina out and look at a few different ganglion cell types. And so these are all of the different ganglion cell types, the output cells of the retina that are sensitive to motion. So they differ in their dendrites. These beautiful branches that you're looking at are their dendrites. They differ in what parts of the brain they send their axons to. And they differ in what aspect of motion that they encode. So we're almost getting there. The cell that I care a lot about, that my lab has spent years studying, is this particular ganglion cell. And this cell we refer to as a direction-selective ganglion cell. Okay, so why do we call this a direction-selective ganglion cell? Well, that's because from the following observation. So again, now we're looking at the retina in cross-section. If you record from a direction-selective ganglion cell, we're going to record the action potentials from that cell, and you take a bar of light and you move it in one direction, you'll see that this cell fires lots of action potentials at the leading edge of that bar and at the trailing edge of that bar. You take that same bar of light and you move it in the opposite direction across those photoreceptors, and this cell fires very little. Okay, so the photoreceptors can't tell what direction the light has gone. They don't talk to each other. So they're just, getting activated when that bar of light crosses their worldview. But somehow 2 synapses later, this cell can not only tell, that the bar of light was moving, but it can also tell what direction it's moving. And so we want to understand how you build a circuit that can detect the direction that something moves. Okay, and that's why we refer to this as a direction selective cell. So the way we represent our data about to quantify how direction selective a cell is we'll plot the number of action potentials that are fired as a function of the direction that the bar moved. So when the bar moves to the right, I think that's your right, I'm not sure, to this direction, it fires lots of action potentials. When the bar moves in the opposite direction, it fires very few action potentials. So if we plot the number of action potentials as a function of direction, we take a vector sum, we get this arrow, and the length of that arrow represents how strong the tuning is, and the direction that arrow points refers to what we call the preferred direction of that cell. So this cell prefers rightward motion. Okay, so and then the opposite direction we call the null direction. So we're going to talk about preferred direction of the cell. Okay, so a really important experiment was done in the 1960s, actually in the optometry department here at UC Berkeley. And these were experiments that were done in rabbit, where they were able to record from hundreds of ganglion cells in the rabbit retina by recording from its optic nerve. And then they played bars moving in different directions. And they found that although rabbits can sense motion in all directions, it turns out that the direction selective ganglion cells, their preferred directions fall along they cluster along 4 directions. So there's actually 4 types of these direction selective ganglion cells: those that prefer left, those that prefer right, and those that prefer up, and those that prefer down. So those are 4 types of these direction selective ganglion cells. So whatever is the circuit we're going to talk about, it's got to be built 4 times, at least 4 times. Okay. So we have, as I said, so early on in the 1960s, when a lot of these visual phenomena were discovered, experiments were done in rabbits, but we have moved to doing experiments in mice. And it turns out mice also have 4 types of these direction selective ganglion cells. And a big advantage of using these mice is that there are transgenic mouse lines in which a fluorescent protein is expressed in, say, let's say there's a purple fluorescent protein in the purple cell, in the rightward preferring cell. There's another mouse line that will have a fluorescent protein in the leftward preferring cell. And then we have another mouse line for up and for down. So the mouse lines are shown by these like HB9, GFP. That's just a nomenclature we use. And so that allows us, when we're doing our experiments, to record from these cells. We can actually find them in our microscopes because they're expressing a fluorescent protein, and we can distinguish them from many others. And the fact that there are mouse lines that you can label them independently really indicates that these are 4 distinct cell types that encode motion in a lot of different directions. Okay, so how do we do our experiments? So the way we do our experiments, we don't do them in living mice, but rather we euthanize mice, and we can dissect out their retinas. So the retina is like this cup, that's at the back of the eye. And then we can take it out and we can make these kind of cuts in it and flatten it down like this and put it in a chamber. And in that chamber, we can perfuse it with solutions that have the right ions and pH that mimic what the retina would have if it were still in the animal. And oxygen, it will be oxygenated. And so those neurons within the retina can still function very well outside of the animal, okay? We now take this this retina that's got this fluid that's keeping the neurons very happy. We put it under a microscope, and we use a special kind of microscope that's called a two-photon microscope. Yaron Nagy has done a lot of beautiful work in developing a lot of microscopy methods. So you've probably heard about two-photon microscopy from her. I will say that the advantage for us to do two-photon microscopy is that it's based on using infrared light in order to stimulate the fluorescent proteins inside of these cells. And by using infrared light, we're less likely to bleach the photoreceptors. So that allows us to hunt and look for our cells that we're going to record from without bleaching out all the photoreceptors. We have also customized our microscope so that we have a visual stimulus that we can project down onto the retina, activate the photoreceptors, and have our drifting bars moving across the retina so that we can record the responses. Okay, so this is all happening in vitro, but the retina works pretty well even when it's outside of the animal. I should say I'm open for taking questions now if anyone has a question or whatever. I don't know, Ron, what's the tradition? Do people take questions at the end? I'm just going to go ahead. Ask a question if you want it. Okay, the other way we use two-photon imaging is to do something that's called calcium imaging. So what I just described is a way for us to find the cells so that we can put our electrodes on them. But there's also a fluorescent imaging method that we can use that allows us to look at the activity of neurons. And that method, again, is called calcium imaging. And again, Nagy is one of the world leaders in developing fantastic ways of doing calcium imaging. And this allows us to look at the activity of neurons. And so calcium, what happens is when a neuron gets excited and it fires action potentials, calcium goes rushing into the cell. And when calcium goes into the cell, we have put a fluorescent molecule in the cell that when calcium binds to it, it'll have an increase in fluorescence. So it's a way to look at neurons that are excited. And so we can also do that to look at direction selectivity. So these are experiments that are being done by Alex Tiriac, who's a postdoc in the lab. So he'll flatten out a retina, he'll put a calcium dye in that retina, and then he'll image, let's say that's a field of view, and he'll take a bar of light and he'll traverse it across that field of view. He'll have the bar of light come in 8 different directions, and he'll map out the responses of that cell. And so when he does that, these are what some of his calcium responses look like. So we can think of this as a burst of action potentials at the leading edge and a burst of action potentials at the trailing edge. And this cell clearly prefers motion up, right? So it has a bigger fluorescent transient when it moves up. And so we think it's firing more action potentials moving up, it prefers up. Okay, so he can do this one cell at a time, but the great thing about doing calcium imaging is now we can record from all of the cells in our field of view, and you get beautiful movies that look like this. So now this is the surface of the retina. You're looking at all the different ganglion cells, all 60 different types, a subset of which are motion sensitive. This big structure here is a blood vessel that's on the surface of the retina. And these cells are glowing because they have a calcium dye that's called Cal 590. And so you'll see now a bar of light is moving across the retina, and these are the responses of these neurons to this bar of light. So hopefully the movie is playing smoothly. I think I should, well, anyway. And so you can practically imagine what direction the bar is moving by seeing as the order in which the cells light up. Okay, so when Alex does an experiment like this, he'll go back and process the images, and he'll figure out which are the cells that are direction selective. And this is a summary of what he's seen. So all the blue cells that you see here are all the direction selective cells. And then we also take advantage of these mouse lines that label, say, one directional preference. So these pink cells are the direction selective cells that prefer downward. And we know that because they're genetically expressing a protein, and we know that that only happens in the cells that prefer down. So the pink are the down cells, the blue are all of the direction selective cells, and then the greener cells are retinal ganglion cells that are responsive but not direction selective. Okay, so we can do this, and by doing this, Alex can walk across the retina, and he can find all the direction selective ganglion cells across the whole surface of the retina. He gets beautiful signal to noise. He can see all the different preferred directions of these cells. And just to show you kind of a summary of what he sees. So what I don't have time to talk about is about how the preferred directions of these cells actually change depending on location in the retina. If you guys are interested in that, I'm happy to talk about it afterwards. But the coordinate system that the 4 directions align to actually are defined by optic flow. And we can talk about that later. For the purposes of this talk, I'm just going to focus on this one location in the retina where the 4 subtypes of direction selective ganglion cells are roughly perpendicular to each other. And so a summary of all the direction selective, the preferred directions of all the direction selective cells are shown here. What's shown in red are the ones that are genetically labeled. So we know that they prefer down, and the green are ones that are genetically labeled. We know that they prefer left. So they provide, I really hate this phrase because I'm talking to physicists, but what biologists will call some sort of ground truth, that we know what the directional preference of these cells are because of their label. There is no ground truth in biology as far as I can tell. Okay, so if you summarize Alex's data, he can find, this is called the Rose plot. It's just a histogram, a polar histogram distribution. You have a lot of cells that prefer left, up, down, and right. Okay, so that's the direction selectivity maps. All right, so that's where we are. So now I've described direction selectivity, but now we want to get at how do you build a circuit that allows you to encode for that. So this is the same picture I showed you before. You record action potentials, bar moves to the right, lots of action potentials, bar moves to the left, no action potentials. So what we want to understand, if we know that the photoreceptors can't tell what direction the bar is moving. So it's going to be something between the neurons that are getting inputs from the photoreceptors and then providing the inputs to the ganglion cells that are actually doing the computation. So some of these neurons in between the photoreceptors and ganglion cells are excitatory onto the ganglion cells, and some are inhibitory. And so we want to see which of these kind of cells are contributing to the direction selective response. So there's a method that we have, a recording method, where you can switch the configuration of your amplifier and look at the strength of those synaptic inputs. So what you're going to see are what the synaptic inputs are that cause that action potential firing. And at the top, you'll be looking at inhibition, and at the bottom, you'll be looking at excitation. And so when the bar of light moves in the preferred direction, you have, this is a lot of excitation. So this is the current generated by excited current, excuse me. And then you have, and then you have a lot of inhibition. You have some inhibition. Now the bar of light moves in the opposite direction. And you see that you get just about the same amount of excitation, but you get a lot more of inhibition. So this is the model of how you get direction selectivity. It's from inhibition. When the bar moves in the null direction, you have a lot more inhibition, a lot more inhibitory neurotransmitter, and that prevents that ganglion cell from firing as many action potentials, right? So the same amount of excitation is there, but this cell is integrating excitation and inhibition. And the inhibition is strong in the null direction, and that's what prevents the cell from firing action potentials. Okay, so we know that we know it's inhibition that's providing, that is essentially breaking the symmetry, and that is giving us direction selectivity. And we actually know the cell that provides that inhibition, and it is this beautiful cell I'm showing you here that's called the Starburst cell. So we know from a series of experiments that I won't tell you about, but they're along the lines that we can transiently make the cell unable to respond to anything, and we can watch direction selectivity go away, meaning that a direction selective ganglion cell, which normally preferred, say, this direction, would now respond equally to motion in all directions. Okay, so we know this is the cell. And the cell is really, it's a really different kind of neuron. Its cell body is here. It actually releases neurotransmitter from the outer tips of these crazy processes that are coming out. And you can see why it's called the Starburst cell. But it's a very symmetric looking cell, right? So, so how did we know, I should say, how do you know that this is the cell? So there are some experiments I didn't tell you about, but I should give you the rough way you do it. The first thing you look at what we call the anatomy. So these are these cells. The purple is the direction selective ganglion cell. The green is the Starburst cell. And if you look at them, if you take a confocal image of these so that you can look at them in 3D and flipped on the side, you can see that this cell physically makes connections, right? So the processes of that Starburst cell are physically touching this ganglion cell, and so you need to have that as a minimum requirement for it to form to prove it forms a synapse on it. The other thing you can do is you can functionally determine it by doing what's called a paired recording. So you can record from a direction selective ganglion cell. You could put another electrode in a Starburst cell, and then you can excite that Starburst cell. And when you excite that cell, you can look to see if it generates a synaptic response in the second cell. And so it does. And so that's the evidence that they're functionally connected via a synapse. Okay. So Starburst cells are super interesting cells because I still haven't explained where the computation comes from, but we know the cell that's providing it. And it turns out Starbursts themselves prefer motion, and the kind of motion that they're sensitive to is outward motion. So this is an experiment that was done by Anna Vlasits while she was a graduate student in the lab. And what she did is she filled the Starburst cell with a calcium dye, and then she imaged out at the very distal tips, which is where the neurotransmitter is released. She took a bar of light and she activated the photoreceptors, that are above this amacrine cell, so stimulates the amacrine cell, and the bar of light moves in that direction. She images out here. She sees a big change in calcium, which we know means the cell's very excited. That same bar of light moves in the other direction. She gets a very small response. So the Starburst cell is sensitive to motion, and it's also direction selective in that it prefers outward motion over inward motion. This is an example of what one of those little synaptic release sites looks like. These experiments were done also by Ryan Morey, where he went on to show that you can do it if you have kind of a bullseye of stimulus coming out, that you get very strong direction selectivity at all of these output sites. So it really prefers outward versus inward. Okay, and the different colors just show you the directional preference, and it's really it's remarkably radial. Okay, so they prefer outward motion. But still, how does that give you direction selectivity, right? So we know we have this starburst cell in the middle. It prefers outward motion. We know that it provides the inhibition that makes those direction selective cells not respond to stimuli moving in the right direction. So the hypothesis is the only way that this cell can do it is if it makes very selective connections to the right direction selective ganglion cell. So the blue process is only going to form connections with a blue ganglion cell where that's its preferred direction, right? So it's going to release a lot of inhibition for this kind of motion. And then and that's going to inhibit motion in that direction. So that's its preferred direction. And this process is only going to form connections with this kind of direction selective ganglion cell, and this process with this type. And when you draw a cartoon like this, it seems so easy, right? So you have kind of a blue molecule that recognizes the blue cell, and you have a purple molecule that recognizes the purple cell, and you can just wire it up like that, right? And so you can have this what we refer to as this asymmetric wiring. The problem is there's a green cell that's sitting right there, and there's a yellow cell that's sitting right there. So, and that starburst cell is really close. In fact, it's touching those processes, but somehow it has to choose the blue cell. And that is a really hard problem. And to give you a sense on how hard that problem is, I want to show you exactly how many starburst cells there are. So the starburst cells form a very thick plexus. So these are all the cell bodies of the starburst cells. Again, we're looking in cross section. If we now look in the flattened view, that's how many starburst processes there are. And this is the ring of the dendrites of that direction selective ganglion cell. And I can tell you all of those processes are touching that ganglion cell. Yet there has to be this incredibly selective. It can only, any given process that's coming out to the right has to only synapse onto a cell that is, prefers the left. Okay, that's a crazy hard problem to do. But it turns out that that's exactly what it does. And the evidence for that is twofold. So one bit of evidence was there was a landmark paper that was done by Kevin Brigman, Moritz Helmstetter, and Winfried Denk. Winfried Denk, some of you may know, he was a physicist who actually invented two-photon imaging, and he was one of the star students of Watt Webb. And they did this landmark experiment where they did calcium imaging to find all the direction selective ganglion cells in the mouse retina. And they did this very high resolution microscopy method that's referred to as serial EM. It's a beautiful paper, but I don't have time to go too much into the methods, but they could reconstruct the dendrites, and then they can see synapses. It's high enough resolution. It's got the 10 angstrom resolution or even better to actually visualize a synapse. And then they found all the starburst inputs to those cells. And this is a summary of what they found. So this is the perspective from the starburst cell. So remember the starburst cell, and they found that those release sites only form synapses where essentially the red ones only form synapses with the ganglion cells where that's its preferred direction, and the yellow only forms synapses where that's its preferred direction. So it's not perfect, right? So this process will hit some greens and it will hit some purples, but it primarily is able to just find the correct postsynaptic cell. So despite this incredibly hard problem of finding exactly the right process to synapse onto, despite the fact you're touching all of them, it seems that that's what they do. The way that my lab sort of got at this question to show that there was this asymmetric wiring was by doing recordings. So again, this is the paired recording where you stimulate the starburst cell and you record the inhibitory synaptic inputs onto the ganglion cells. And if that starburst cell is located on the null side, you get a big synaptic conductance. But if that starburst cell is located on the preferred side, you get a small one. And so our physiological experiments also showed there's an asymmetry in the connectivity of these starburst cells on there. Okay, so this is it. I've now spent a long time telling you what the problem is, and now I'm going to tell you how we're trying to solve it. So the problem is that you have a motion-sensitive neuron that prefers outward motion, and the way that confers direction selectivity onto these 4 types of direction selective cells is through this very precise wiring. Okay, so the question that my lab is trying to figure out is what instructs this asymmetric wiring? How do they do it? How is this achieved? Okay, and so the first thing when you're studying the development of any circuit is you want to know, when does it occur? So the first thing we want to know is, does the animal have to see? Like, does the animal have to have any visual experience in order for these direction selective cells to wire up? And we're going to go back to Alex's method for this. And what he found, so this is the direction selectivity, if you remember, the distribution of the preferred directions in the adult retina. And when he repeated this experiment in a 2-week-old mouse, which is the age at which a mouse opens its eyes, so when it has its first visual experience at 2 weeks old, it looks pretty much like the adult, right? So we see robust direction selective cells. If you look at their distribution across the retina, it looks incredibly similar to what you see in the adult, meaning that we think that you don't need visual experience to set up direction selectivity. Okay, so it's set up before vision. So when is it set up? So now we can't use a visual response to figure out when it first emerges, because as soon as there's a visual response, it's already there. Like, it's already wired up to do it. So as soon as there's a visual response, the circuit is already there. So how early does it get set up? So for that, to answer that question, we go back to those paired recordings that Wei did. I should say Wei was a postdoc in the lab when she did these experiments, and she's now a professor at the University of Chicago. And what Wei did while she was a postdoc is, she found that the cells on the preferred side had strong synapses, and on the null side, if the Starburst cells on the null side, it was on the preferred side, it had weak synapses. So strong inhibition, weak inhibition. And she did that right when the eyes open, and the asymmetry was as strong right when the eyes open at postnatal day 14 as an adult. But then when she did it 1 week earlier, when the animal was 7 days old, it turns out that the Starburst cells were symmetrically and weakly connected to direction selective ganglion cells. So it was something that happened between postnatal day 7 and postnatal day 14. And actually, Ryan Morey, who is a graduate student in the lab, actually pinned down the date to be around between postnatal day 9 and postnatal day 11. There's a 2-day period of time when this asymmetry emerges. And what's happening during that time is that's a period where there's a tremendous number of new synapses forming. And we were able to show that this was actually due to new synapses forming. So there were synapses here, but they were weak and symmetric. In this 2-day period of development, all of a sudden, there's a whole bunch of new synapses that form, and they only form when that Starburst cell process is oriented such that it finds its right postsynaptic partner. Okay, so we know it happens in a short time during synaptogenesis. All right, so I still haven't told you anything about what instructs this. And I'll tell you right now, we don't know the answer, but we have a really surprising result, and this is what I'm going to end with. So what instructs this asymmetric wiring? So whenever you ask a question on how something gets wired up in the brain, mechanisms for development get categorized as either nature versus nurture. So the question is it genetically programmed? Everyone's expressing like the right, surface molecules that recognize each other. Oh, I'm sorry. I have a dog that howls when sirens go by. Can you guys hear it? It's my dog howling. Sorry.
No worries.
All right. He's done. He's not
Is
done.
it
Okay.
is it direction specific?
Yeah, and actually it's a great example of directions. Like, so,
Outward
sorry about
versus
that.
inward.
Okay, so nature versus nurture. Actually, it turns out there's direction selectivity in the auditory system for frequency chirps. So there are cells in your auditory system that will respond to increasing frequency chirps versus decreasing. So direction selectivity is not just in the visual system. Anyway, nature versus nurture. Is it genetically programmed, or is neural activity involved? Well, I just told you this gets set up before there's any vision. So what would be the neural activity if there's no vision at this age? And so we're going to take a slight diversion, even though I told you we're almost done, into the other big thing that my lab studies, which is this phenomenon called retinal waves. So, and we're going to talk about why we think retinal waves are involved in setting up direction selectivity. And this is work, again, being carried out by Alex and a new graduate student in the lab, Karina Bystrong. Okay, so before the eyes open in a mouse, this is when the direction selective circuit is getting wired up. I just want to talk a little bit about how the retina develops. So mice are born with their eyes closed. Their photoreceptors are actually not very mature. And importantly, the synapses between the photoreceptors and the ganglion cells don't exist. So these neurons that connect the photoreceptor to the ganglion cells don't exist. Light is not affecting these ganglion cells. But it turns out the ganglion cells are not silent at this age, but they're spontaneously active. So again, we're doing a calcium imaging experiment where we're going to look at neural activity. In this case, we dissect the retina in, we put it in our chamber, and we just take a movie. And we just look, we're not applying any light at all. And you'll see these beautiful spontaneous increases in calcium that we know are these cells are spontaneously firing action potentials. And if you look at a lower magnification, so you're looking at a square millimeter of the retina, you see this beautiful spontaneous propagating activity. And this is what we refer to as retinal waves. And my lab has spent, since I was a postdoc, I've worked on this phenomenon, trying to understand how it's generated and what role it plays in development. And so we know a lot about the circuit that generates retinal waves. And one of the things that's very interesting about it is the circuit that generates it actually involves that starburst cell and a direction selective ganglion cell. So they're both involved in generating the wave. They both get depolarized by the wave. And interestingly, the synapse that connects them has this particular protein. We won't go into the details of what it is, but happy to talk about it later, that this particular protein is critical for generating the waves. And if you knock out the gene that codes for that protein, you actually can get rid of waves. Okay, the other thing I want you to know about waves is that they actually have a propagation bias. So although they can initiate anywhere and they can propagate in any direction, there is a big bias for them propagating nasally on the retina. So many more waves go in one. So there's information about direction in the firing pattern. Okay, so what happens when we have a mouse that doesn't have waves? So we're going to go back to our imaging results. So remember, in the adult, there are four, the preferred directions cluster along these four axes. This is, they're present at eye opening. If you look in this mouse that doesn't have waves, it is completely missing horizontal direction selective cells, right? So here we have a lot of horizontal preferring cells, and they're just not there at eye opening. And they never form. Even after this animal has had visual experience, you never get those horizontal preferring direction selective ganglion cells. Okay, so what does it mean to not have, so from this, we think that the waves are important for setting up direction selectivity. So before I talk about how waves might do it, just what does it mean to give you a sense of if you don't have these horizontal direction selective ganglion cells? So it turns out that a long time ago, over 10 years ago, there was actually a study that showed in these same mice that don't have waves, that this mouse lacked an optokinetic reflex. The optokinetic reflex is the reflex that your eyes use to stabilize objects as you move through the world. So, and so your eyes are constantly adjusting as you move through the world it's a reflexive eye movement. You're not even conscious of it. But it turns out that there's that if you don't have that direction selective cells, and there's actually a genetic condition, a monogenetic condition for humans who have it, you have something that's called nystagmus. And nystagmus means your eyes actually jitter in the, and if you have horizontal nystagmus, your eyes will jitter in the horizontal axis. And I couldn't find the movie before this talk to show you, but you can look up nystagmus and see movies of people who have it, so that they can't stabilize their gaze and their eyes are constantly moving. And actually direction selective ganglion cells are important for being able to stabilize your eyes. And there's actually a mouse that is also missing that gene, and it's also lacking direction selective cells. So getting rid of these direction selective ganglion cells in horizontal really affects your visual input. But how do waves wire this up? And this is the beauty, or maybe the horror of biology, is that every time you answer a question, you only start with many more. I don't know why waves set up direction selectivity. So as I said before, they have a propagation bias. So maybe there's directional information in the waves that will strengthen the synapses between the starburst cells and the direction selective ganglion cells. And when you have more in this direction, maybe those synapses will get stronger. But that only explains one type of direction selective cell, not the other type. And then what about the vertical ones? Like, how do they get wired up? Another possibility is that waves are actually just providing changes in calcium or changes in membrane potential that are combined with a molecular marker that's actually providing the information for the wiring. And these are things that we're working on addressing in the lab. Okay, so to summarize what I talked about today, direction selective circuits emerge in the mouse prior to eye opening, independent of visual experience. There is a potential role for this spontaneous activity called retinal waves in the establishment of horizontal direction selective ganglion cells. And just in a couple, unfortunately, things I didn't have time to talk about, we have done some experiments where we did pursue other hypotheses and what we thought was important for setting up direction selectivity. We had mutant mice that had very distorted Starburst cells, so they didn't have a real radial symmetry. We had ones that had very messed up dendrites of the direction selective ganglion cell, and you could really mess up what the shapes of those cells are, and it looks like direction selectivity forms completely normally. And these are two studies that were published in the last few years. And we are looking for these molecular markers that might distinguish the 4 different direction selective ganglion cells using a method called RNA-seq. And so we are also ongoing and doing those experiments. Okay, so let me thank the people who did the work. Sorry, this is the Feller Lab. We're fine. We're all fine during the pandemic. You don't have to worry about us. We're doing great. No, but I would much rather show a picture of the lab in the before times when we all got to get together. This is the Feller Lab celebrating Pi Day. I want to highlight the work that was fun. The people in blue are the people whose work I talked about. I want to thank Matthew Summers. I used many of his slides. He just gave lab meeting, and I used many of his slides for our talk today. Karina and Alex, I talked about their work on setting up direction selectivity, and Malik El-Kesni's whose work I didn't get to talk about. I want to give a shout out to Ben Smith, who is an imaging specialist who works with us to design our custom-made microscopes. We have a couple undergraduates in the lab. These are the alumni whose work I have talked about, who've worked on direction selectivity, and all of our work is funded by the National Eye Institute. So thank you very much.
Thank you, Marla, for the wonderful talk. I see there are some written comments or questions.
Oh, great.
Can you see them?
I can now see them. Okay. So Steve Beckwith asked, so Steve asked this question. I'm sorry, I should read it out loud to everyone.
Read it.
So, when you refer to direction, there must be a direction on the flattened retina. And is there a difference between the actual directions on a live retina compared to the flattened sample? Yes, so this is a very interesting question. And one of the things that I had mentioned that I did not go into detail was is what do those directions align with? So, and I had said that they align with optic flow. And so what I'd like to talk about are the axes of optic flow, right? So optic flow means when you move through the world, as you walk through the world, and you walk by objects, those objects traverse across your retina, right? And they create motion. And that is a stimulus for a direction selective ganglion cell. So for humans, there's this kind of singularity right in the center of our retinas, right, where all those motion vectors come out of, like just as you move through the world. For mice, their eyes are on the side of their head. And so as they move through the world, that point where all those vectors converge is actually in this, like in the very tail end of their retinas. And so you get a lot of distortion of space around that optic flow axis. And so we actually think that the direction selective ganglion cells, actually their motion, their preferred direction follow these optic flow axes. It's better with a diagram, but hopefully, here, maybe I can share my screen one more time just to show you a picture of what optic flow looks like. I think I have it in the talk at the very end. Yeah, so this is like what optic flow looks like for humans. Now for mice, it's sort of at the end. And so when we show this I was showing this grid. Where is it? I apologize for doing this. I hate when people do this. Okay, and I'm doing it. Here we are. So these grid, this is that singularity I'm talking about. For the mouse, it's in the what's referred to as temporal retina, and that's where the converging optic flow vectors go. So Steve, does that answer the question? Right. So that's what defines direction.
Yeah, thanks. You have answered the question. I think I was also just curious because if you show these radial diagrams, they're not exactly symmetric. I mean, they have sort of weird directions, and it struck me maybe the weird directions have almost as much to do with the conformal change you make when you strip the thing down and flatten it as they do with a weird direction that the mouse has to deal with in its brain.
Yeah, so we don't distort it too much, but I do think it, but they do, I will say that they follow those vector flow. I just showed you one area, but the preferred directions will distort more as you get more peripheral and near those points. So, but yes, it's a great question. Okay, let's see. Vintley asked, regarding the result that direction selectivity is caused by increased inhibition, is this the result a general one or just for specific cells in mice? So this is biology. So that is a really, so for the cells I talked about, those particular motion sensitive cells, it is absolutely true. And across species, it's true. There are direction selective cells found in different parts of the visual system. Some are found in cortex, and there it's not true. There is a debate in the retina world about a different kind of direction selective cell, about whether it's inhibition or whether it's excitation or it's some combination of them. So it is a general thing that's true for, but I mean, it's not a general thing. It's a thing that's true for this particular circuit in the retina. But I thought about saying this at the beginning. This is, I will tell you, direction selectivity is kind of, dare I say, the canonical computation that many people in neuroscience have been trying to solve. People have been trying to solve this since the late 1800s, right? There's a lot of behaviors in animals that indicated direction selectivity. Cockroaches will turn in the direction of optic flow. That was the first thing that characterized it. So people have been building models and testing it in many different, I told you, for auditory cues, for animals using their whiskers. They, whisking this way versus that way will give different responses in neurons. Mice that are navigating through a maze, going this way versus this way, will activate neurons differently. So it's like this motif that people are always trying to solve. So for the direction selective ganglion cell, that's the most well described one, and that's why so many people in my lab keeps working on it. It's kind of the canonical computation. If we can figure that, then maybe we can figure out something more complicated. Okay. Vint, did that answer that question?
Yeah, it did. Thanks.
Okay. Okay. Is it Elon Roth? So the pulse in the neuron seems like a large amplitude structure moving in an inhomogeneous medium, preserving its shape. Can it be described like a soliton balanced by nonlinearity, inhomogeneities, etc.? Okay. The action potential is the coolest thing that neurons do. And often people will ask me why I became a neuroscientist, and my answer is because of the action potential. So the action potential is amazing. I can give a whole lecture on the action. I love the action potential. So it is all about membrane charge. It's not, and it regenerates at every point in the membrane. And it can regenerate at these points in the membrane because of this collection of ion channels that are in the membrane that allow that to happen. So it's a regenerative phenomenon. And so it doesn't have it's not that it disperses and it needs kind of the soliton, it doesn't have to have the same properties as like a soliton has. There are some examples of where it has to propagate very long distances, and there's some tricks that the nervous system does that allow it to preserve its shape without decaying, but it's just because it can be regenerated at each point in the membrane that it's able to keep its shape and amplitude. Okay. Okay, how does the starburst cell know to fire more light when the light goes outward from the, it doesn't know anything, but it has, but this is the beauty of neurobiology. So that question, why does the starburst cell prefer out versus in? My lab has published 3 papers on that question. There's about 6 labs who are working on it. It's it's a really rich problem. There isn't a single thing in neuroscience that isn't rich. So I've been telling you about how the synaptic inputs really dictate the computation, but it turns out there are electrical properties within the cell itself that allow it to do that. So essentially you can think of it as the cable. So it's a thick cable that is integrating current for light that moves out, and then it's got a very thin end, and it's truncated at the end. So it has a high resistance end. So you can literally use sort of RC circuit kind of analyses to say, is it going to more effectively give you a bigger voltage change if you sequentially activate this way versus that way? And so, so the answer is it knows it because the electrical properties of the process itself facilitate that direction selectivity. It also gets Starburst cells, it turns out, synapse onto each other, and where those synapses are also dictate why it's direction selective. So there's actually a very deep explanation as to why it's direction selective. So the neuron doesn't have to know anything. It just have to have the right electrical properties that allow that to happen. Okay. Is there any known connection between retinal waves and visual snow syndrome and other entropic phenomenon? I don't know what that is. Do you want to tell me what visual snow syndrome is? Does someone want? This is Rob Cook.
Yeah, it's a rather rare one that actually I have. It's where you see static over your eyes. So like if you tune your television over like an old analog television to static, and you see that over your eyes everywhere. Whether your eyes are open or closed or not, and your pictures of retinal snow look very similar to what I see. So that's why I'm asking the question.
Yeah, I don't know. So some people have told me, particularly the waves that I show, some people have told me that they see that, and apparently they can see that if you push on your eyeballs really hard. So, and so apparently when you push, so this is not answering your question, I apologize, but it's answering some question. And I think that when you push in your eyeballs, you can actually, you can disrupt the blood flow. And if you disrupt the blood flow, that pressure, that can make neurons start to fire in this kind of wave-like way. I mean, in your case, do you know if it originates in the retina? Have they told you it originates in the retina? Like, I wonder if it originates in some other part of your visual system.
It is very, as far as I know, it's very poorly understood. And it was only very recently neurologists have gotten together and said, wait, that's a thing. So.
Oh, okay. Oh, I'll have to look it up. Yeah. I'm sorry that you have that. All right.
I suppose you answer all the questions.
It looks like it. Oh, so someone named Edward wrote the direction of the wave. I don't know what the question is.
Yes, what I'm asking is, it was too complicated to write and listen to you at the same time. It looks like the direction of that synchronizing wave, I'm going to call it a synchronizing wave, is from the southwest to the northeast, kind of. And I didn't know if that was just a particular example, or if it's always that way, or more importantly, if there's any connection between the direction of that overall wave and the direction sensitivity of the cell in question.
Yeah, so I will say that there are more cells that prefer the nasal direction, and there are more waves that propagate in that direction. So the idea is a little bit of the following, to give a kind of evolutionary argument for waves. So frogs, vertebrates, but not mammals, they don't have waves. And when they're born, they swim. And when they swim, they have this optic flow, which is sort of stimulating across the retina. So again, the lens inverts it, so it's moving kind of nasally on the retina. So it's motion kind of in that direction for optic flow. So mammals they have these waves. In humans, they're fetal. In mice, they're born, but they're not moving. And so the thought is that these waves, they have a propagation bias that is in the same direction that would be generated by optic flow. And so the idea is, well, maybe, I mean, this is speculative, but maybe this is a reason that they have that propagation bias. But the question is, and that might be the reason why direction selectivity is able to be established sort of I just made this whole argument about how it lines up along optic flow. Yet, how does an animal know what optic flow is if it hasn't moved and it hasn't had any visual experience to tell you what those axes are? And so maybe the waves are giving information about optic flow before the animal actually is experiencing it. And that might be there to sort of help set things up. But like I said, it doesn't vertical. So.
But wouldn't that mean our optic flow would be coming from the center of our vision, spreading outward?
Yes, that's what it is for humans.
Oh, that's what it is for us. And then, are there any other directions of flow besides that overall direction, or is that just monotonic? Is there a spectrum of directions?
Well, so maybe not from our motion. Well, certainly when you move, there's all sorts of.
I don't mean for us. I mean for. I don't mean for us. I mean for the mice.
Yeah. So if you looked at that movie that I showed of the GoPro on the head of the mouse, then there's a lot of different directions that the mouse sees due to its own motion. It rotates its head. It's rolling around. It's moving forward. So there's a lot of different.
Yes, but that's what I'm referring.
A really interesting question about for that visual neuroscientists are trying to understand, how do you separate, like, how do you know what's motion on your retina due to self motion, and how do you know what's actually something moving in the world, right? And that is something that your brain has to deconvolve. And we actually think that maybe the direction selective ganglion cells are helping to let you know what's due to self motion, and then that maybe that signature can help you figure out things that are moving independent of you. So there's a lot of problems that need to be solved. Yeah. I think that maybe it's time to go, Edward, but if you'd like to stay afterwards, we can talk.
I'm referring to that synchronizing wave that comes across before they're born.
Yeah. Yeah, those go in all directions.
Okay, so is there a preferred direction?
Yes, there's a preferred direction that is the same as the optic flow direction.
Okay.
Yeah.
Thanks.
So I suppose there's no more question, or?
Oh, wait. Oh, yeah, there is some more.
Oh.
Lots of compliments on the talk. We don't want to not read those, but I'll skip those for now. Okay. So Daniel, I can't quite see the names. I apologize. Is it iBot? Thanks for, okay, what breaks the rotational symmetry of the direction selective ganglion cells? How does a particular cell type that prefers downward motion know which direction is down? So they don't know anything. What they know is it's down because that's the, it's getting input from that starburst cell that's going to release more inhibition for upward motion. Right, so it doesn't know anything. It's just getting inhibited for upward motion, and so therefore it's going to prefer down. Right, so I think it's because of how it's wired into that circuit is what conveys its direction selectivity. Does that answer the question?
Yes, I think I misunderstood that you had said it was actually a different, actually a different type of cell, but you're saying it's just from the interaction with the neighbors.
Yeah, from who's synapsing onto it. But it is a different type of cell because somehow it's only getting input from the Starburst cells that, inhibit up. And it's not getting input from the Starburst cells that inhibit left. And so it's got to be different in that it recognizes those differently. But and I have to say, these are some of the questions. Do they all start the same? And maybe when you get all the nasal ones first, then the other ones become temporal. I don't know. These are a lot of questions that we have to, but we do think that at this point, we're kind of thinking there really are 4 different cells that are very, very similar, except that they get that inhibitory input from a different oriented starburst process. So crazy.
Thank you.
Yeah. Well, Marla, okay.
I'm still here.
Okay, good. So you answer all the questions, I think, right?
Great.
Okay.
Yes.
So thank you so much. It's a wonderful, interesting talk.
Thank you. I see some old friends. Are we allowed to stay on the Zoom just a little bit longer?
Sure, why not?
Okay.